Today we are going to discuss about the Phage Display Library. Usually the library is of antibody, so most commonly it is called Phage Display Antibody Library. As you can see here, this is the phage particle. It has DNA, this is genetic material, and it has the code protein. So what scientists have done, they have fused the code protein with the antibody fragment while inside this vector, and when this virus is expressing this code protein, that is also expressing the antibody fragment. So, in short, the phage virus is displaying the antibody fragment when it is being fused with the code protein. This the phage is displaying the antibodies along with its code protein. So this is the code protein, this is the antibody fragment. But whatever the modification we will do at the genetical level, not at the protein level. So one more time, this is the phage virus that is expressing the antibody fragment along with its code protein. So when the code protein or the genes for code protein is fused with the genes for your protein or the target gene, then this virus is going to express your target protein along with its code protein. So this is the phage display library. Usually and most commonly it is used for the antibodies, so it is called as phage display antibody library. So as you can see here, the fusion of a specific protein or a peptide, because antibodies are proteins, with a code protein on the surface of filamentous phage particles for the selection. There are many methods which are being used by the phage for the phage display library, but the most common methods are using either the phage vector system or we are using the Phagemid vector system. So when you are using the phage vector system, so in this case, it allows the expression of the code protein to the foreign protein or the antibody fragment. In this means the foreign protein is what is in this case it is antibodies, and they are driven by the natural phage promoter. So this vector, this contains all the genes. So this vector contains all the genes. So this is the vector, and this vector, the phage vector, and this is your target protein. Let's say this is your target protein or antibody. In this case, it has all the genes which are necessary for the replication and the packaging of the virus or the virions. This vector has all the genes which are necessary for the replication of this vector or the genes, and this vector also contains the genes for the packaging of the virion. So this is itself sufficient. It doesn't need any kind of helper phage virus when you are performing the phage display library. So additional helper phages are not required for phage packaging with phage vector system. But there are some limitations. This is the multivalent display, means here we can show that this is the polyvalent phase. So this code protein, which is so let's say this is the code protein, the black portion. This is your target protein, which is your antibody. So there are multiple copies of a single antibody. So in case of the phage vector, you are getting the polyvalent or the multivalent display, means more than one copy of the foreign protein of interest per phage particle. So what it will do, even though if it is having, like, let's say if it is having multiple copies of your protein, and if you want to isolate a antibody against your antigen that is having a high affinity, then in this case the phage vector system is not a good vector system because there are multiple copies. So even though there is less affinity, then also due to the avidity, means the strength of the receptor ligand complex, which is the antigen antibody complex, is quite high because multiple copies are present. There are multiple copies. If there is a single copy, then it is okay. But since there is a multiple copies of a single protein of interest, even though the affinity for your target antigen is less, but due to the avidity, you will get a more affinity. So this is the disadvantage of this if you are using the phage vector system. The another thing is this: when you are using the phage vector system, you have to be quite careful that you are not disrupting any important genes which are necessary for its replication and the packaging of the virions. Okay, so these are the two limitations. Again, I will tell you, because they are having multiple copies of a single protein, even though the effect or the affinity for your affinity towards your target antigen may be less, but because there are multiple copies, when you will perform the experiment, you will find them higher affinity because this is the multiple copies. This is due to the avidity. This is the major limitations when you are using the phage vector system. And the advantage is this, that you are not using any helper phage virus because this vector is self-sufficient for doing the replication of viral genes and the proteins. In the Phagemid vector, there are two origin of replication is there, like the plasmid origin of replication is there, and the filamentous bacteriophage origin of replication is there. So this vector can grow in bacteria. And why this filamentous bacteriophage origin? Because it is there, so single stranded DNA could be packaged into the virion. But to produce the single stranded DNA, let's say if this is your single stranded DNA, but to produce the single stranded DNA, you need multiple enzymes along with the core proteins, which this vector doesn't have. So you have to provide the helper phage virus, and this helper phage virus will provide everything, like the enzymes that is needed for the production of the single stranded DNA. So that is the thing. So you have to use the helper phage virus when you are performing your experiment with the Phagemid vector, because that will help in the production of those proteins which are required for the production of single stranded DNA. And that single stranded DNA is going to be packaged into the virions. So this is the thing. So this is the Phagemid vector that is having the antibiotic resistant genes, the plasmid origin and the filamentous origin. So helper virus will not be having this origin of replication, so it is inefficient in self packaging. So if this is your helper phage, because it doesn't have origin of replication, which is 1AF, then it is inefficient in self packaging. So self packaging is not there, but when you will bacteria will be having this plasmid vector, and when you will transfer that bacteria with this helper phage, then you will get the packaging of the virions, and you will get the your protein of interest along with the core protein. So once it is there, you will transform into the E. coli cells. You will super infect with the helper phage. Now the helper phages will provide all the enzymes which are required for the replication of the single strand or production of the single stranded DNA, and that DNA is going to be packaged into these virions. So here you can see that this is the Phagemid virions, that is having the code protein, and this code protein will express your target protein. So this is regarding the Phagemid vector. So two things you have to remember: it has two reasons of replication, and when you are performing your experiment, the antibody library, the helper phase is needed. And why it is needed? Because it is providing all the enzymes or the code proteins necessary for the replication or the production of the single DNA so that it could be packaged. This is the Phagemid vector system. So here you can see it doesn't carry the entire phage genome and thus require the helper phase for the phase packaging. The expression of the antibody P3 protein by Phagemid system requires an artificial promoter such as the LAC promoter. It will only allow the monoblend display, only the single copy. If we talk about here, you will only get the single copy of the protein. If you prefer multivalent copy, then you have to use the P8 protein. You have to fuse your target gene with the P8 gene. If you prefer the multivalent display, it would be better to use the modified helper phases or display the foreign protein fused to the P8 protein. So two code proteins are important in this phase virus. One is the P3 and another is the P8. If you are using the Phagemid vector and if you want the multivalent display, means multiple copies of the same protein, then you can fuse your target gene with the P8 protein or the P8 genes, or you can modify the helper phase. Plasmid is more efficiently packaged than the helper phase DNA. Most of them contain the plasmid inside. A minor fraction would contain the helper DNA inside. When you are superinfecting these cells with the helper phase, so chances of packaging of the helper DNA is less because of its large size. So most of the virions will contain a plasmid DNA. So that is the thing. And the minor fraction may contain the helper phage DNA, but since this is large, it is difficult to package. Here you can see the comparison. This is the phase vector, this is the Phagemid vector. This is the P3 protein. Again, this is the P3 protein. You have your displayed sequence along with it, and when you will transform in the E. coli, you will get the phage virus or particle that will be having the code protein, and that will show the multiple copies of a single protein. Here we can see the multiple copies of single protein, and here you will get single copy. So let's say if you have the antigen and you want the antibody that is higher affinity, so you should go for this one. Okay, because if you go for this one, this will anyway shows high affinity. Whether it is having the higher affinity or not, it will show the higher. So this is the concept of the polyvalent and the monovalent phase. There are multiple types of antibody library. The one is the naive library. This is being made by using the donors which are not being exposed to any kind of diseases by your knowledge. And what you can do now, you can take the variable region of the heavy chain and the variable region of the light chain and the linker region, which is the glycine and serine rich region, and you can just insert that genes in the Phagemid vector. You will transform in the bacteria, and then you will infect with the helper phase virus, and then you will get something like this. This is the heavy chain of the variable region of the heavy chain. This is the variable region of the light chain. This is the glycine serine rich region, and you transform in this bacteria, super infect with this helper phase, and then you will get the virus particle that will be displayed. This is the code protein, the yellow portion, and you will be getting the antibody fragment, which is a single copy of antibody fragment. But now let's understand this in a bit more. What you are going to do, let's say you have 100 of donors, let's say you have 100 of donors. Okay? What you will do, you will just isolate the RNA, convert that cDNA separately, and then just mix it, make it up. How many donors you can take? The library size will depend on the number of donors you are going to take. If you are increasing the number of donors, the library size is going to be more. Usually the preferred size is 10 to the power 8 to 10 to the power 10. Means it has these many antibody molecules. So it has these many binders for the target antigen. Larger the library, it is better for sale, it is better for your experiments and all. So this once you make this cDNA full, you have to amplify the variable region of the heavy chain. So, and how you can do that? There is a website VBase. You can also click on this link, and you will find the primers against the variable region of the heavy chain or the light chain. You will get all the primers. How many sequences are there? You can get it down. Usually there are 8 primers that I have known till now. So you will amplify these variable region of the heavy chain, and once you have this gene, let's say this is your gene, you will add the restriction site here in your primers. Okay. So this is, let's say this is your cDNA. It will be having the variable region of the heavy chain. You will use the primers, forward and the reverse primer, and you will be having in these primers the restriction site. Okay. These are the restriction sites you will be having. So now you have this vector, which is the PCENT. You can use other vectors as well, and then you will digest these vectors with the same restriction enzymes. Let's say if it is like SCAI, like not one, then you have to digest this vector also with this SCAI or not one. Okay. So you have to just study this vector, how many sites it is having, and once you know this is having these restriction enzymes, you can just design the primer that is having these sites for the restriction enzymes. Then you will just add them in the forward primer. Let's say this is your forward primer. Let me just show you. You have designed the primer, which you can see here, this one. Let's say this is your forward primer, and then you can add somewhere it will be having the SFI sequence, which is the restriction enzymes. Like this, you have to design your sequence. Once you will amplify these genes, you will be having the restriction site as well as the variable region of the heavy chain. Then you will digest the vector with the same restriction enzymes. You will ligate them. Okay. And once you will ligate them once you will ligate them, then you have to decide the insert to the plasmid ratio. Okay, that is going to give you the best transformation. Then you will perform the transformation reaction. So once you have, let's say if you have this plasmid DNA, which is restricted, means it has been digested with the restriction enzymes, then you have your cDNA, the PCS, the amplified sequence that is having the variable region of the heavy chain and contain the restriction enzyme site. Then you will ligate it by doing just simply the ligation. And after doing the ligation, you will do the transformation in the bacteria. While performing the transformation, you have to be sure how much ratio you can use. Like you can try with 1:2, 1:3, like 1 this, 2 this, 1 this, 3 this, or 1 this, 4 like this. And based on this, you will do multiple transformation, and you will see how many cases you are getting a higher number of the transformants, which is having your product, which is having your ligated product, means that is containing your variable region along with the plasmid DNA. And once you found, let's say if you found, okay, 1:2 factor is going to give you the best ligation, then you will perform a multiple transformation, because you need to increase the library size. So you have to do, like, let's say 40 electroporation you can do in the bacteria, and then those bacteria you will just select those colonies which is having your ligated product and make a pool and keep it up. Here you have to decide your library size as well, and you can decide the library size by counting the number of colonies, the dilution factor, and the initial dilution. Like, let's say you got the 1000 colonies and the dilution is, let's say, 500. Initial dilution, the stock was 5 ml, and you take the 100 microliter. So it will be how much you can decide, 25 into 6, which is 2.5 into 10 to the power 7, or you can say 0.25 into 10 to the power 8, which is not good. So this is not good. So you have to do then again multiple transformation. Let me just rub it on here. So actually what I told you, first you will go to this website. First you will go to this website, design the primers, and based on the vector which you are choosing, if suppose the vector is having the restriction site for these two enzymes, then you have to include these two sites or the sequences that code for this restriction site to your forward and the reverse primer, so that you can digest with the same restriction enzyme this amplified sequence, and as well as you can digest the vector with this amplified sequence, and then you will take the 1:1 dilution or 2:3 dilution, like you can take the plasmid in one factor and your amplified product 1 microliter, you can take your plasmid 1 microliter, take your product 2 microliter, and see in which you are getting a higher number of transformations. Because once you are doing the ligation, you will do the transformation. You will select the colonies which are having your desired gene of interest, and that you can do by some restriction digestion or by sequencing. And once you are confirmed that you are having the heavy chain of your variable region of the heavy chain, you can select that transforming. Decide the library size, and the library size, as I told you, how you can decide it by, like, let's say if there are 1000 colonies, or let's say 10,000 colonies, if there is a 500 factor of dilution, and let's say initial stock is 5 ml, you took the 100 microliter, then it will be like 25 into 7, which is like 0.25 into 10 to the power 9. So this is the good library size, because the library size which I can say here is 10 to the power 8, 10 to the power 10 is going to be a good library size. If your library size is going to come less, then you have to do multiple electroporations. Once you have done the PCR amplification and you have select the colonies, then you can just freeze it, make the glycerol strong, and just keep it at -18. Similarly, you have to do the reaction for the variable of the light chain, and the light chains are two types of light chains, copper chain and the lambda chain. Both you have to perform separately, and then you have to amplify the linker region. And linker regions from where you have to select the clone that is having the linker sequence, which is the glycine and the cysteine region. Then you amplify that sequence, and then again you have to ligate. While you are performing the ligation, you have to be sure that both the things are having the same restriction site. So your ligation, like for your linker, if it is having the let's say SCF-NOT1, this is just an example. So then your this factor should also contain the same restriction site, so that that can be ligated with that. So what is the point here is, first of all, you have to go to this previous site, select the primers for the lambda and the kappa chain, do the amplification by using the primer that is containing the restriction site, ligate them. Then again you have to do the transformation and select the colonies which are having the desired sequence, desired gene sequence, which is the variable region of the light chain. And once you have done that, you need to amplify the linker region from a clone that is containing this site and ligate them with the pool of V lambda and V kappa. So if I'll tell you here, if you will perform this experiment, this is going to be very huge. So what, let's say you have cDNA. Okay. You amplified the variable region of the kappa, variable region of the lambda by using many primers. Like I can show you how many primers are there. Like you can say for the V lambda there are V lambda there are 11 primers, V kappa there are 6 primers. 17 reaction you have to put the pool. It's going to be a tedious one actually. Once you have done with this P kappa and V lambda, then the ligation part is done, then you will do the transformation separately for V kappa and V lambda. This separate transformation you will do, and then you will select the colonies which is having the V kappa, and then again you will select the colonies which is having the V lambda. Okay, and once you are done with a selection of these colonies, you can just preserve it. Glycerol stop. Okay. And keep it, and again you have to be sure that the colonies, the library size should be like 10 to the power 8 to 10 to the power 10. If it is less, then you have to do multiple transformation. This is a very tedious process. If you want to make a library, this is going to be a quite tedious process. And once you have done this part, then you will take the clone, SCFP clone, which is having the linker region, then you amplify this region, and then you ligate, and then you have the colonies, right? And then you will just pull out the colonies. Like if suppose there are these many colonies which are having your desired gene V kappa, and here there are colonies which are having the gene for your V lambda, let's say this is V kappa, then you will just pull it up. There is no separate you need to do the glycerol. You will just pull it up here and pull it up. And then after you are done with this, you will just pull both of them, V kappa and V lambda, and then you, whatever the sequence you have, you are amplifying for the SCFP linker, you will just ligate these genes with the plasmid DNA that you will isolate from the pooled colonies that will be having the V kappa and the V lambda. Going to be quite tedious process. There are multiple ligation, there are multiple. First of all, there are you have to collect multiple donors, make the RNA, make the cDNA. Then you need to do multiple PCR amplification. Then you have to do multiple ligation, many transformation, and then selection. And then again, once you are having this the colony for the VH library, you have the linker VL library. Then you can simply mix them and do the pull through PCR. The pull through PCR is the PCR, again you are adding the restriction site here by using the primers, and then you will ligate them with this vector. Again you will do the transformation, and again you will going to select the colony, and then again if the library size is less, then you will do the multiple electroporation. As far as I know, minimum of 100 electroporations are needed. Minimum. You can go for many more if your library size is not good. So this is about a library. In short, we'll say we will amplify the fragment of the variable region of the heavy chain and the light chain and the linker region, and then we will combine them. That will form the SCFP. SCFP means it's a library that contains only the variable region of the heavy and the light chain. So here if you see for the VH repertoire, you will see we have the 8 primers. For the JH, we have the 4 primers. Then we have to do how many reactions? We have to each reaction, because for the GI we are going to make a pool of these primers, and for each set of the primer you will use one set of VH and the mixture of the GH primer. So total are going to be the 8 reaction, PCR reaction. Then we will cool it up, then we will ligate, then we will electrophorate, then we will select the transformants which are containing the VH feature. Similarly, we are like in this, I said 8 primers, 1, 2, 3, 4, 5, 6. So basically, it is like 9 primers. 9 primers. And the JH are the 4 primers. So 9 reactions you have to keep. And here you can see we have added the restriction site. And in this, we are adding the JH1, XH1 site. The plasmid we are going to digest with the SF1 and the XH1. Amplified product we are going to digest with the SF1 and the XH1, so they will like it. Similarly, for the VL repertoire, here you can see for the V lambda we have 11 primers as counted, and 7 is right, and 11 primers. For the V kappa there are 6 primers. So total number of reactions are going to be 17 reactions. So the process is like very laborious process. Instead of I'll say technical, it is very laborious process. Similarly, for the V kappa, there are 6 primers, so you can count there are 6 primers. Then A lambda and J kappa, these are the primers which we are going to use. And this is, we are showing how to introduce the linker into the pool of your V kappa and V lambda. Let's say if you have the V kappa, V lambda library, you will make a pool. You will just combine the V kappa and V lambda, and you will ligate your SCF linker. So this is regarding the naive library, and the immunized library is same like the naive library. The only difference is that we are immunizing the animals, or we are collecting the blood from the person or the patient who are being infected with the disease. For example, like, in case of the coronavirus, you can see if the person is infected with the coronavirus, we can make the COVID library like that. And the other things are quite same. Another library is the synthetic library. In this, we are not collecting any blood or something. Everything is going to be done in the laboratory only. Here you can see this is the bacteria, and that is the DUT and the UNG negative. So what we DUT is UTPase. So this is the DUT negative and UNG negative. So because of this inactivation of this, UTP is going to be accumulate, means the uracil is going to be accumulate. So in some point of this replication, you will find there are U, and because this is a UNG negative, this U is not going to be removed from the strand. So this bacteria that is having the vector, and this vector is having the U instead of T. Now you know that in the antibody region, let's say this is the antibody, this is the heavy chain, this is the light chain, and this is the heavy chain. So in the heavy chain or the light chain, only the variable region, and in the variable region basically CDR1, R2, R3, these are responsible for binding to the region. Specifically CDR3 is majorly responsible for binding to the antigen. So you will introduce the mutation in the CDR3 side and design the primers, and then you can just ligate them in a vector that is containing the CDR1, CDR2 or CDR3 sequence. So once you will do the amplification, elongation by using the polymerase or the ligase enzyme, that will produce the second strand, because it's a single strand template. This is a single stranded DNA. So the next strand will be having your mutated DNA. Right? Now you will do the transformation in the UNG positive bacteria. Means now the uracil is going to be degraded so that your initial strand. So this is going to be removed. Now your second strand is there. Then it will replicate. Then you will get the double stranded plasmid here, and then you will transfer the super infect this bacteria with the helper phase, and you will be getting the library that will express your mutated oligonucleotide. oligonucleotide. So specifically what scientists are doing, they are introducing the mutation in the CDR3 region. Here, everything is being done in the laboratory. There are lots of modifications have been done in this, but this is just the basic. In this is a bacteria that is having the plasmid, and this bacterial strain is DUT negative or UNG negative, and because of this, there is a accumulation of the uracil, which is not going to be removed because of the UNG negative portion. The UNG means uracil and glycosylase. So uracil is not going to be degraded in this, while DUT means this UTPase, and because this is like negative, so there is an accumulation of the UTP, and you will get the U in the single stranded DNA. Now you will introduce the mutated oligonucleotide. From this bacteria, you will isolate the plasmid. You will get this your mutated oligonucleotide. You will just mix them up, and then you will transform in the UNG positive bacteria super intact with the alpha phase, and you will be getting a virus particle that will display your gene of interest. So here you can see this is the VH library that you make, the VL library that you make. This is the bacteria that is containing a VH library. This is the bacteria that is containing a VL library. You will isolate the plasmid, so that will be having a VH library, VL library, like this. Then you will introduce the mutation restriction site, which is the pull through PCR. Then you will transform in the, you will ligate them in the vector, and then you will do the transformation super infect with the virus alpha phase, and you will be getting a virus particle that will display the protein of interest. This is regarding the antibody library, and we will discuss next lecture the logistic regression.